everyone. Welcome to Science for High School with Merivit Education Center. My name is Master 11 and this series is for Math and M.3. Now, we will discuss the topics all about inheritance of traits and genetic disorders. Here are our objectives for this lesson. First, the students will be able to explain genetic inheritance from the combinations by considering the only feature that the dominant allele completely dominates the recessive allele. Second, students will realize the changes of genes and chromosomes that may cause genetic diseases and give example of genetic diseases. And lastly, students will realize the benefits of the knowledge of genetic disease. Now, may I ask you something? Do you have any ideas on where do our traits came from? Why do people have blue eyes while the other have black ones? Why do you have pointed nose while the other have shorter and wider? Different measurement of height and blood type. Why do some people do have straight hair while the other have the curly ones? Do you ever think and question about that? Well, my dear students, it is all because of the inherited genes you get from the chromosomes of your parents. The traits that you possess are combinations from your father's genes and your mother's genes. Genes are the basic unit of heredity located on chromosomes. They are short sections of DNA responsible for the transmission for the characteristics from parents to offspring, like possessing different colors of eyes, straight or curly hair, height, and etc. Inherited traits are passed from parents to offspring according to the rules of Mendelian inheritance. We have here Mr. Gregor Mendel as the father of genetics. Through his work on pea plants, he discovered the fundamental laws of inheritance. He deduced that genes come in pairs and are inherited as distinct units, one from each parent. The Mendel's laws of inheritance include the law of dominance, law of segregation, and law of independent assortment. At this moment, we have here how you are able to acquire the genes of your parents. We all know that humans do have 46 diploid number of chromosomes, right? Do you have any idea on what is chromosome? Chromosomes are made up of DNA, genes, and proteins. It is a condensed unit of DNA. Your entire genetic code are represented by chromosomes. You inherit 23 chromosomes from your mother and then 23 chromosomes from your father. That will result to 46 diploid number of chromosomes with 23 pairs. Each pair have one chromosome from your mother and the other one is from your father. Each pair of chromosomes that you acquired from your mother and father contributes an allele, which is a variant of a gene. It is a variety of a gene or a form of a gene. It is either the same form of a gene or different form of a gene of a specific trait. A pair of your chromosome is consist of one allele from your mother and one allele from your father. That will result to a combination of genes from both parents. Now, let us go deeper into allele. An allele controls the traits of an individual. Each trait consists of two alleles. It is either dominant allele or recessive allele. A dominant allele is a variation of a gene that will produce a certain phenotype, even in the presence of other alleles. The dominant allele will show its effect when it is present, and usually a capital letter is used to represent the dominant allele. On the other hand, the recessive allele can only show its effect in the absence of dominant allele, and a small letter is a representation for the recessive allele. Alleles are an alternative form of a gene on a homologous pair of chromosomes at a particular location called gene locus. Gene locus is where the different genetic code is found. Those genetic codes are the traits being inherited from your parents. In addition, Homologous chromosome is a pair of chromosomes with same gene sequence, loci, chromosomal length, and centromere location. As what we can see in the diagram, during meiosis, the homologous pairs of chromosomes are separated, leaving only one allele of each trait in each gamete. Each offspring receives one allele of a pair of genes from each parent. Moreover, we can also see in the diagram that both parents are homozygous in pairs and have their own the same traits. In the case of the mother, we have here a pair of both dominant alleles or a genotype of homozygous dominant, which has a phenotype of curly hair, while the father possesses two recessive alleles or a genotype of homozygous recessive that has a phenotype of straight hair. So, as a result of combination of two alleles from both parents, in which the mother provides the dominant allele while the father provides the recessive allele. After fertilization, the genotype of the offspring will consist of a combination of one dominant allele and one recessive allele, or what we call heterozygous. Similarly, as there is the presence of dominant allele, then it will typically show its presence and produce certain phenotype even if there is the presence of the recessive allele. Likewise, we call the genotype of the offspring heterozygous as it has a combination of two different alleles and will have a phenotype of curly hair. In addition, the organisms that do possess two alleles that are the same is what we call homozygote, while on the other hand, heterozygote is an organism that possesses two different alleles for a same trait. A while ago, I have already given you the same example of diagram that what we have right now. However, we will just focus on genotype and phenotype to make it clearer. Well, genotype is an individual's collection of genes or genetic makeup of an organism composing two alleles inherited for a particular gene, while phenotype is the individual's observable traits from the expression of the genotype. In this case, referring from the diagram, the offspring has an inherited trait which is curly hair that was the result from the genotype pair of one dominant allele and a recessive allele, or what we call heterozygous in genotype. Referring from our previous diagram again, the table on the screen shows clear differences among the combinations of genotypes of alleles that will result to a certain phenotype. So, if the genotype of the offspring that has a combination of dominant allele and recessive allele, then the genotype of the offspring is heterozygous with a phenotype of curly hair. Next, if the genotype of the offspring is homozygous dominant, then its phenotype is still curly hair. This is because of the presence of both dominant alleles. Lastly, if the genotype of the offspring is homozygous recessive, then its phenotype is straight hair. This is because of the absence of the dominant allele. Now, we will proceed on how we can predict genotype and the phenotype of an offspring easily. And a monohybrid cross is an example for that. A monohybrid cross is a genetic mix or a hybrid between two individuals who have homologous chromosomes that are homozygous in genotypes, or genotypes that have both dominant alleles or having both recessive alleles, which result in opposite phenotypes for a certain genetic trait. In the sample diagram, we have the genotype of parents, which one of them is homozygous dominant while the other is homozygous recessive. During meiosis, as the homologous chromosomes are separated in the gametes, as we cross it out, we can see that all the possible genotype of each offsprings are heterozygous. The dominant trait will basically expressed as the dominant allele is masking the recessive allele. In this example, we need to refer from the given informations in which we have here the dominant allele capital letter H represents the curly hair and recessive allele small letter h represents the straight hair. Now, this example is a cross of phenotype with both curly-haired parents which are homozygous dominant genotype. As the homologous chromosome separates in the gametes, we have the result that both alleles in each gamete are dominant, and basically these pairs of dominant alleles will be passed on to offspring that has a result of 100% probability to have a genotype of homozygous dominant. And similarly, all the possible offsprings will have phenotype of curly hair. Now, we have here another way on how to cross and predict probability of the genotype and phenotype of an offspring. A Punnett square is a graphical representation of the possible genotypes of an offspring arising from a particular cross or breeding event. Creating a Punnett square requires knowledge of the genetic composition of the parents. The various possible combinations of their gametes are encapsulated in a tabular format. Therefore, each box in the table represents one fertilization event, and always keep in mind that each box in a Punnett square is worth 25% of probability of the genotype and phenotype of an offspring. Going back to the example that we had a while ago, we will try to predict the probability of the offspring's genotype and phenotype using the Punnett square. We all know already that we have both parents' phenotype, which is curly hair, from the genotype of each parent that are homozygous dominant. As we draw our Punnett square, it shows that it has a 100% probability that the offspring will have a curly hair phenotype, as all of the possible pairs of alleles in the Punnett squares are all homozygous dominant, which has the same result from the crosses we did a while ago. Another example that we have here is a cross between homozygous dominant parent and heterozygous parent. So, as we cross the homozygous dominant to heterozygous, it shows that the genotype that we have in the crosses create two homozygous dominant and two heterozygous, or have a genotype ratio of 2:2 for two homozygous dominant result and two heterozygous, or you can also put 1:1 for the genotype ratio, for it will represent the homozygous dominant and heterozygous. Likewise, the phenotype of all the possible offspring will still have a curly hair because all the crosses possesses at least one dominant allele. Likewise, when we cross the homozygous dominant parent and heterozygous parent using the Punnett square method, we will also have the result of having two homozygous dominant and two heterozygous, which will result that all of the offsprings will have a genotype of curly hair because of the presence of dominant allele in each pair. This confirms that the results of our both crosses are the same and correct. Another example, we have here a cross between homozygous dominant parent and homozygous recessive parent, which we will still refer from the informations that capital H represents curly hair and small letter h represents straight hair. You know already how to cross it, right? Okay, very good. As what you can see in the diagram, when we cross homozygous dominant parent and homozygous recessive parent, we will have four sets of genes which are all heterozygous. In this case, we have a result of 100% probability that we will have a heterozygous genotype of offspring, and basically the phenotype of all the possible offsprings will have a curly hair. Similarly, as we use the Punnett square method, we can see in our Punnett square table that we also come up with the same results of genotype, which are all the possible offsprings are heterozygous, and will also have a result to a phenotype of curly hair. Last example is a cross between two heterozygous parents while referring from the same information. As what we can see, as we cross two heterozygous genotype of both parents, we can see that in the result, there is one homozygous dominant, two heterozygous, and one homozygous recessive genotype. Then basically the genotype ratio of the cross is 1:2:1, which will be what you expect for a typical monohybrid cross. While when we look onto the phenotype, its phenotype ratio is 3:1, which shows that 3 among of the possible offspring will have a phenotype of curly hair, while the other one will have a phenotype of straight hair. Likewise, when we do use the Punnett square method, we can observe that we do have the same result with the cross we did a while ago. We do have one homozygous dominant, two heterozygous, and one homozygous recessive, which will result to a genotype ratio of 1:2:1, while the phenotype ratio is 3:1, which means that out of 100%, there is 75% probability that the offspring will have a phenotype of curly hair and 25% will have straight hair. As of this moment, we have learned already on how our traits were being inherited. Now, we will proceed on the genetic disorders that might occur during inheriting those genetic traits. Do you have any idea on what is genetic disorder? A genetic disorder is a disease caused in whole or in part by a change in the DNA sequence away from the normal sequence. Some causes of genetic disorder is the inheritance from the parents. Some are from environmental exposure, such as chemicals and radiation. One example of a genetic disorder is color blindness. Color blindness is a minor disorder. It is a problem for a person who has it, but certainly not one that limits his movement or shortens his lifespan. Most people who suffer from color blindness cannot differentiate red and green. More males than females are colorblind, as color blindness is linked to sex. Next, we have here another example of genetic disorder, which is what we call hemophilia. Hemophilia is a medical condition in which the ability of the blood to clot is severely reduced, causing the sufferer to bleed severely from even a slight injury. The condition is typically caused by a hereditary lack of coagulation factor. Another genetic disorder is the sickle cell disease. Sickle cell disease is a group of disorders that affects hemoglobin, the molecule in red blood cells that delivers oxygen to cells throughout the body. People with this disease have atypical hemoglobin molecules called hemoglobin S, which can distort red blood cells into a sickle or crescent shape. Another genetic disorder is the Down syndrome. Down syndrome is a condition in which a person has an extra chromosome. Chromosomes are small packages of genes in the body, right? They determine how baby's body forms and functions as it grows during pregnancy and after birth. Typically, a baby is born with 46 chromosomes, while the Down syndrome has an extra pair. Another genetic disorder is thalassemia. Thalassemia is an inherited blood disorder caused when the body doesn't make enough for a protein called hemoglobin. Hemoglobin is an important part of the red blood cells. It is caused by the changes to the DNA of the cells that produce hemoglobin. Hemoglobin is the substance in the red blood cells that allows them to carry oxygen. A thalassemia patient has less hemoglobin and fewer red blood cells in his body. The low hemoglobin and fewer red blood cells of thalassemia may cause anemia, leaving patient fatigued. As of this instance, we are done already with the different types of genetic disorders. Now, we have here a way on how to prevent or reduce genetic problems. Premarital screening is a group of tests recommended for couples that are going to get married to prevent as much as possible transmitting diseases to their offspring. Premarital or preconception tests includes testing for genes infectious in blood transmitted diseases. This will also help to reduce the spread of some heritable diseases and some infectious diseases. This is the end of our lesson about inheritance of traits and genetic disorders. Thank you for listening and see you on the next episodes. Goodbye.